Today I'm going to speak about a new era in the infant nutrition, which is called HMOs. My presentation will contain historical review, prebiotic and probiotic terminologies, and human milk component and HMO structure, and then we will talk about studies which have been done on the human milk oligosaccharide. And historically speaking, in 1899, the Henry Tischer isolated bifidobacteria from the breastfed infants, and he found it abundant. So he said those breastfeeders are more abundant in this bacteria, so it could be related to something secrets or some secrets ingredients in the breast milk. And then in 1930, the characterization of the first individual HMO was described, and in 1950, the 2FL structure was discovered. So the 2FL, our guest today, which I am going to talk about it, it was initially identified in 1950. In 1965, Lilly and Stewell give a definition of a probiotic, and we will see later what's probiotic. And then in 1995, the definition of prebiotic was introduced, which is the non-digestible ingredients that selectively enhance the growth of the beneficial bacteria. And in 2016, they update the definition of prebiotic, and in 2018, the launch of HMOs. Now, what is prebiotic? To understand HMOs, we should understand what is a prebiotic, what's a probiotic, what is symbiotic, what is postbiotic. All these should be understood to understand then what is human milk oligosaccharide. So simply the prebiotic is a substrate, this is the updated definition, a substrate that's selectively used by the host microorganism to produce a health benefits, usually in the form of oligosaccharide. What is the characteristics of prebiotic? It is resistant to digestion in our GIT. It goes from the up to being secreted in the stool as undigested, and then fermented by the gut flora and protect against attachment and invasion of the organisms and increasing the beneficial bacteria. Then, if we can see in this study simply that the bacterial flora in the intestine, before starting the prebiotic, the predominance was bacteroids, 65%. And after giving 15 grams per day of prebiotic to those volunteers, after 30 days, the bacterial flora changed to 71% of bifidobacteria, which is the beneficial bacteria. So this is what's called prebiotic. It means enhance the growth of beneficial bacteria. Okay, prebiotic is available in many sorts of foods, and some companies manufacture some forms of prebiotic to mimic the human milk oligosaccharides. What's a probiotic? Is a living microorganism, when administered in adequate amount, confers a health benefit to the host. It could be used orally. Now there is a vaginal suppository to decrease vaginosis and many other uses for the probiotic. And mainly it is in the form of lactobacillus, which is present abundantly in the yogurt. All the yogurt is lactobacillus, and also in the bifidobacteria. Symbiotic, it means any product which contain both prebiotic and probiotic. It's called symbiotic. And postbiotic is a metabolic byproduct produced by the probiotic and make some metabolic changes or benefits to the host. If we go to the start of the day zero, or when the babies are delivered, the GIT is relatively sterile. But when at the age of 30 days, we can compare the breastfeeders with the formula feeders. The microbiota is different. Bifidobacteria is a dominant in the breastfeeders, while in those who are bottle feeders, it's usually equal. So more the bifidobacteria, the beneficial one. Anyhow, whatever we are doing or trying to do, we cannot copy the human milk. Maybe the macro structure of the milk could be copied, but the micronutrients, it's very hard, very difficult to produce it. Maybe nucleotides added, lipid and fatty acid, DHA, arachidonic acid, some are added, but it's very hard to copy all these micronutrients, all the benefits which are present in the breast milk. For that reasons, the breastfeeders having less lower respiratory tract infection, all the trials showing that, and less risk of diarrhea. And also there is a low morbidity for the short terms and low morbidity for the long term, like obesity and type 2 diabetes mellitus, and also increased intelligence in those who are breastfeeders. Now, grossly simply, when we look to the composition of the breast and cow's milk, there is, we can find that the third structure or component of the human milk is the human milk oligosaccharides. The third component, while in the cow's milk, it's a negligible amount. So lactose, fat, and then oligosaccharide, it's also more than the protein composition. So this is the grossly. And when we look to the different mammals, we can find that elephant and humans having very large amount of oligosaccharide, but the goats and cattle and cows having negligible amount of oligosaccharide. This is what's called species-specific variability. Now, let's go to the composition of these oligosaccharide. We can find that there are different types of oligosaccharide. The terminology is a little bit complicated. It's not like a simple galacto-oligosaccharide or fructo-oligosaccharide. We will see that the monosaccharide which is present in the human milk oligosaccharide is different. So here we can see that the 2-fucosylactose is the most abundant form of human milk oligosaccharide. It represents nearly 30% of the human milk. Let's go back to the what is oligosaccharide. First, there is a monosaccharide, which means there is one molecule, simply glucose or galactose or fructose. This is an example, of course, of monosaccharide. And what is disaccharide? It's either lactose, which is called sugar milk, containing glucose and galactose, sucrose, what's called fruit sugar, which is glucose and fructose, and maltose, 2 glucose molecules. This is called disaccharides. And oligosaccharide is 3 to 10 molecules of monosaccharide. This is called oligosaccharide. Usually it is non-digestible again. Our digestive system cannot digest the oligosaccharide. Now, this is the 5 building blocks. It's like Lego. When you to build a Lego, it's a 5 blocks. Mainly glucose, galactose, and acetylglucosamine, fucose, and sialic acid. This is the 3 main building blocks. And usually the core of the oligosaccharide is the lactose, glucose, and galactose. And this will be attached to either sialic acid or fucose. So when we'll see here, the next slide. If it's attached, we call it fucosylated amino acid. Oligosaccharide. Fucosylated oligosaccharide. And it represents 41% of the human milk oligosaccharide. If the fucose is not attached, it's called non-fucosylated and represents 47%. Both these two, we call it neutral human milk oligosaccharide. If the sialic acid are attached, which is only 12%, they call it sialated oligosaccharide. Lactose. This is lactose, glucose and galactose attached to sialic acid. They call it sialolactose. Okay. So now Let's go to the simple one, the trisaccharide, 2-fucosylactose. The 2-fucosylactose is the simple form and most abundant form of human milk oligosaccharide. Fucose attached to galactose and glucose. The human milk oligosaccharide varies from time to time. In the colostrum, it's very high, 20 to 25 gram per liter. And usually during the lactating period after the colostrum, it is from 5 to 15 gram per liter. But also it varies from individual, from mother to mother. On the same session even of the feeding, it could be varies from diurnal variation from the morning to the evening. The amount is different. Okay. Also the mother's nutritional status. The mothers, 80% of the mothers are secretors of the 2-fucosylactose because they express the enzyme which is called alpha-1,2-fucosyl transferase. And this enzyme catalyzes the addition of a fucose, okay, to the HMOs in the lactating glands. So 80% of the mothers are contain 2-fucosylactose and 20% will not excrete 2-fucosylactose. When the human milk oligosaccharide enter to the GIT, it have a different action from the stomach until the colon. First, in the stomach and intestine, it's to prevent the prevention of pathogen adhesion, and the same will be done on the intestine and the gut maturation and surface glycosylation, which is very important, and in the colon, the fermentation of the gut microbiota and enhancing the growth of the beneficial bacteria, and this is what's called bifidogenic effect or a prebiotic effect. And there is nearly 1% or less than 1% are excreted in the urine. For that reason, it's believed that human milk oligosaccharide have a systemic effect because it's absorbed, partially absorbed in the blood, and it have some systemic effect, and as we will see in the next slide. So 2FL coming up, down, no salivary, no pancreatic enzymes, no intestinal enzyme can digest it. Okay? And in the colon, as I said, it selectively enhances the growth of the beneficial bacteria. And also, on the fermentation process in the colon, it produces the short-chain fatty acid, which it helps in the energy, gives some energy and also regulate the contractions of the colon. As we will see in the next slide, it is interact with the epithelia and reduce the inflammation and may change the surface carbohydrate profile. Okay. In vitro studies demonstrate that 2FL reduce inflammatory cytokines released by immune cells. In this slide, we can see how the HMOs improve the host defense. It's directly interact with the intestinal epithelial cells and modulate their glycan expression. Glycan expression in the mucosal cells, if there is any microorganisms, it needs a receptors to be entered to the mucosal cells. If the glycan expression changes, there will be no receptor, so it will be not invading the mucosa in the intestine. Okay, and here the HMO prevent pathogen adherence to the cell surface by acting as a receptor decoy. Decoy, it means it will be on the surface, the mucosal surface, and it catches it as a receptor, but it is not another receptor, so it will avoid it from internalization or entering to the mucosal cells. In this study, we can see that the comparison of the study was to investigate the effect of maternal secretory status. The woman who secrete 2-fucosylactose okay, on the development of breastfed gut microbiota with a special emphasis on the bifidobacteria. So there is, as I said, there is a secretor woman and non-secretor woman. The microbiota are different. Even if she is a breastfeeder and non-secretor, the bifidobacteria more in the secretor's mothers. The mother will secrete 2-fucosylactose, having more bifidobacteria and less Clostridium and less E. coli than those who are non-secretor. Here we can see that whenever the fucosylactose are increased, there is a low risk of the infection with E. coli. Whenever the secretion of 2-fucosylactose are less risk of the infection with the E. coli. And the HMOs protect against diarrhea. It is found that there is, whenever the high concentration of the 2FL, there is a low risk of the Campylobacter diarrhea, and also low risk of all causes of diarrhea whenever there is more concentration of 2FL. Now let's go to the growth and morbidity. This is very interesting study. A cohort study. I'm sorry, this is another slide. In 2006, the breast, this is a very nice also study, Stefan, 2006. It shows that higher concentration of HMO was significantly associated with decreased risk of respiratory and GIT illness. Now, this is the study which I talk about it. They make a cohorting of two groups. One group taking a regular formula and the other group taking another formula fortified with 2FL and another human milk oligosaccharide. And those who are finishing the studies who are taking the formula fortified with human milk oligosaccharide are 57, and those who are in the control or group are 64 infants. And the endpoint was to check is there the safety of the human milk oligosaccharide, the growth, and the morbidity. And they found that the morbidity are low in those who are taking the formula fortified with human milk oligosaccharide, and they found that bronchitis risk decreased by 70%, and the lower respiratory tract infection decreased by 55%, and the antibiotic need decreased by 55% also, and the antibiotic need was decreased by 56%. So what's new? Fortifying the formula with the human milk oligosaccharide is safe, the growth is okay, they have a low risk of morbidities. This is what is new. This study is also interesting. There was a trial on the giving a rats. They found that the rats fortified with this form of oligosaccharide is called decyclolacto-antitrose. It could be protection of the necrotizing enterocolitis. So they make a trial. They collect 200 women with very extremely low birth weight who are more liable to develop necrotizing enterocolitis, and they put a hypothesis that those mothers with a low DSLNT oligosaccharide could be a risk factor to develop necrotizing enterocolitis. And the result, 8 infants in the cohort developed NEC, and all those 8 infants having their mothers having a low concentration of the DSLNT. So in conclusion, this form of human milk oligosaccharide could be a predictor of the patients who develop necrotizing enterocolitis, and could be a tool to the future prevention and treatment on the necrotizing enterocolitis. For the allergy, it's well known now to talk about human milk oligosaccharide and the risk of allergy. It's well known that the breastfeeders have a low risk of allergy, and in this study we can see that they make a comparison of the breast milk profile of the HMOs in those who will develop the cow's milk protein allergy. They do a challenge test at the age of 6 months to see who developed cow's milk protein allergy. They found a profile, those who are having cow's milk allergy having low profile of 4 types of human milk oligosaccharide. And then those who are having late onset of cow's milk allergy, their mothers are a secretor of 2-fucosyl-lactose, and those who are having early type of cow's milk protein allergy, their mothers are non-secretor of 2-fucosyl-lactose. Now the relation to the cognition, most of the studies showing that the IQ of breastfeeders are higher than those who are non-breastfeeders. They do a test of fortifying the milk to the rats and to the mouse. And those who are taking 2-fucosyl-lactose having higher excitatory postsynaptic potential in the rats and in the mouse than those who are not taking the 2-fucosyl-lactose. So oral 2FL improved electrophysiological process in the brain devoted to natural communication, to neural communication. Again, 2FL increased hippocampal long-term potential in vivo during infancy. Those who are taking 2FL having high excitatory postsynaptic potential. And 2FL, not the fucose. Now here they give fucose alone and 2-fucosyl-lactose. They found that those who are fed 2-fucosyl-lactose specifically having higher excitatory postsynaptic potential. Here the 2-fucosyl-lactose and here control and fucose alone. What is interesting here also when they do vagotomy to these rats, they found that the effect or the benefit of the 2-fucosyl-lactose are stopped after vagotomy. And here we can see that the rats with vagotomy, the excitatory postsynaptic potential are low. But those taking 2FL with no vagotomy, still the excitatory postsynaptic potential are high. So the benefit of 2FL on the CNS function are dependent on the vagus nerve integrity, suggesting gut-brain axis plays a role in 2FL-mediated cognitive benefits. So now when we talk again briefly on HMOs, we have many benefits, starting from the prebiotic effects, decrease the risk of adhesion and intestinal epithelial cells modulation, as we said, expression of the glycan, immune modulation, and modulator of this leukocyte rolling and adhesion, and probably there is a role of the 2FL in the brain development and in the IQ. The take-home message, the HMOs are